Hi, everybody. I think we're going to get started on the competition portion of the symposium. So my name is Michelle Arkin. I'm at UCSF in the Small Molecule Discovery Center. And I am going to introduce the competition. This is the CEND target competition. I'll introduce the judges, a little bit about drug discovery, and then the last year's winner, a colleague of mine, Larissa Podust, and then this year's contestants. Okay, so starting with, the goal of the CEND target competition is to select new targets and new target biology. People present their desire in their project to do high throughput screening, and the award actually is to do a high throughput screen in our lab. So it's nice, and it's really great. And so far we've had two winners that have run screens with us, and they've both been really high quality screens, really high quality collaborations, as you'll see when Larissa presents her results. Okay, so Tamina gave me 5 minutes to introduce the process of drug discovery, and I figured that that's fine. I can say everything I know about drug discovery in about that amount of time. But I figure there are two ways to give the process of drug discovery, and one is over time, and one is within a project. So those are the two slides. So over time, what's a brief history of drug discovery? So we started out up until the 1960s with a phenomenological approach. Natural products, sticking compounds into animals, seeing what happens to them, and this was really pretty successful. And the way my colleague Adam Renslow, who runs the chemistry group at the SMDC, describes it is this is the age when we were actually doing drug discovery, and what we do now is really drug invention. So now drugs are compounds that never existed in the universe until they were synthesized by a medicinal chemist, and gone, sadly, largely, are the days when Fleming could just find something on his bench that solved a major problem. After we had this age of phenomenology, we started learning a lot about biochemistry and molecular biology, and now we entered an age of about 20 years that I like to call the age of rationalism. Very mechanistic-based biochemistry, structural biology, modeling, so computer modeling of what proteins look like and what protein active sites might look like. We have molecular biology, which ushers in the age of biologic drugs, so just give people the proteins themselves instead of some analog of the protein. And now we're in the age of parallelism. So an age where we can do things really fast, really cheap, the $200 genome, the ultra high throughput HTS, and where we have 3,456 experiments in one 4x6 inch plate. So this is high throughput screening and ultra high throughput screening, and this is because we have robotics that allow us to miniaturize experiments, and this is what we do in our lab. We also have combinatorial chemistry, which allows us to make a lot of compounds, put a whole lot of compounds into those dishes very quickly. And now biology is kicking in with the omics era of biology, where we can take a systems-wide approach and sequence the whole genome or exome. And we're hoping now that the information that we learned didn't really help us get drugs all that quickly. Maybe just throwing a whole lot of samples at the problem will get us from target to drug more quickly and efficiently. So that's yet to be seen. We'll see. The other way to look at drug discovery is within a project, what are the different steps that you go to from a target to a pill? And we can divide this into 9 steps. Here we have target ID. So doing novel biology in the laboratory, we identify something that has a really important impact on the therapeutic area that we're studying. We then have to turn that target biology into a hit, so a compound or a protein that has some of the biological effects that we're interested in. But you can see that that's really just way up here, and this is what high throughput screening addresses, is this dark green chevron. After that, we have hit to lead chemistry, where now we're not optimizing based on the initial biological discovery, but based on a host of in vitro properties that give us some idea that the compounds that have that biological effect will also have that effect in a more complex organism, first a cell, then an animal, then a human. Lead optimization is really optimizing for all of those pharmaceutical properties in the context of an animal. And then IND-enabling studies are largely toxicity and safety studies that tell the FDA that it's safe to put it into patients. And then we have 3 clinical trials, and finally FDA approval. So along this pathway, 1 in 10 projects gets through this stage, to this stage, and I think what's really shocking to a lot of people in basic biology is that only 1 out of 10 compounds gets from this stage out the bottom. So this does continue to be a very steep funnel. And all these things tell us that there's still a huge need for innovation in lead discovery and in drug discovery. Okay? And that's really a function that we can tackle in an academic setting. The cost of drugs, as we discussed this morning, is really extraordinarily expensive. It takes a very long time. And then you have the patent life of the agent that you've been developing, and that patent life is starting to waste away during that time. 1% of projects reach the NDA, that was the number I just gave you, and 70% of those fail in the clinic, mostly in phase 2 clinical trials. So after a lot of work and love and brain power has gone into these things. So this tells us we just don't really understand biology well enough, and the interaction of small molecules with biology. And then finally, it's worth noting in the omic age that all drugs target about 200 out of the 20,000 gene products, or 500,000 different types of proteins with all the post-translational modifications that we have. Only about 200 of them are targeted with drugs that are out today towards humans. Then there are a set of antibiotics as well. So it's my belief that academic research can help fill this innovation gap and lower the risk of new targets by understanding those new targets better, and new technologies by understanding how molecules work and how molecules interact with proteins better. Okay, and that is my history of drug discovery and the process of drug discovery. And now I'm going to introduce the judges, and then I'll introduce last year's award winner. So the 3 judges that we have judging the competition today, we'll start with Gideon Bolag. He completed his PhD in biochemistry from Berkeley, from this lauded institution. He then was worked at CEDUS, then was a founding scientist at Onyx, where he worked on the project team with Bayer that led to the development of a drug called Nexivar, which is an anti-cancer agent, a kinase inhibitor. And now he's the vice president of research at Plexicon. Kevin Giudice did his PhD at UCLA. He's a chemist, medicinal chemist. He was a postdoc here at Berkeley. And then he held various leadership positions at Genentech, where I met him, and at Theravance, where he was one of the starting scientists. At Theravance, he was a team leader for telavancin, which is an antibiotic for multi-drug-resistant bacteria. And now he is the president and CEO of Achaogen. And finally, Joya. Oh, Joya, I don't know how to pronounce your last name. Shaletsky. She got her PhD in biochemistry from Harvard, so she's not from Berkeley. While she was there, she worked with Tom Rappaport in protein translocation channels in the endoplasmic reticulum, and she's currently a group leader at Cytokinetics, which is the epicenter for cell-based screening technologies. Okay. And now I'm going to introduce Larissa. Larissa. Larissa Podust got her PhD in Russia before moving to the United States. She was in the, were you on the faculty at Vanderbilt? Yeah, she's on the faculty in biochemistry at Vanderbilt, and now she's an adjunct professor with us at UCSF, working in the Sandler Center for Drug Discovery, where she's the core director for crystallography and structural biology. And she's going to talk to us today about the project that she did as part of the CEND, as her CEND competition winner in cytochrome 51, CYP51, CYP51 for trypanosomes for T. cruzi.
Microphone. Do you have?
No, it's not. It's just.
Okay. Hi. Once again, I'm a recipient of last year award, and my relationship to Berkeley. I'm Berkeley mom, at least former Berkeley mom. My son graduated Berkeley last year, so at least I justify my presence here. And I think since I presented this work last year, it would be useful if I remind you what it was about. And work was on Chagas disease, which is neglected disease. And this disease is endemic for Latin America, and about 20 million people in these countries are infected with Chagas, have Chagas disease. And this is the main cause of heart failure in these countries. Diseases also emerging in the United States, like Arizona, Texas area, and also in Spain. Causative agent of Chagas disease is Trypanosoma cruzi, unicellular organism that infect heart muscle and over the time of 10-20 years, if untreated, destroys human heart. Current therapy for Chagas disease was introduced in 1960s and suffer from side effects, low efficacy at chronic phase, and also emerging drug resistance. And of course, there is a need to identify new therapeutic targets and develop better and safer drugs. And the target we proposed last year, new target for T. cruzi, is cytochrome P450 CYP51, which catalyzes sterol 14-alpha demethylation in the biosynthesis of membrane sterols in T. cruzi. And if we inhibit CYP51, we basically shut down sterol biosynthesis in this unicellular organism. And what happens then is the following. This is the healthy T. cruzi cell, which has a nicely defined thin membranes, and this white drops here, which store lipids. And as soon as we begin treatment, first of all, T. cruzi exhausts all this internal storage, and then cytoplasm membrane begin to deteriorate, like blabbing appears, and also internal membranes in the cell are also destroyed. Eventually, T. cruzi cell dies. Although this target is new in Trypanosoma cruzi, in fungal diseases, this is very well-validated target and multi-billion-dollar business for pharmaceutical companies. Such drugs like posaconazole, fluconazole, voriconazole, and drug in clinical trials, ravuconazole, all inhibit CYP51 enzyme in fungi. And this drug also interacts with Trypanosoma cruzi CYP51. These are crystal structures solved by my group, complex of between CYP51 and posaconazole. You see that posaconazole molecule is almost too long for this protein, and the tail, which is this part, is wobbling around in the funnel of this active site entrance. This is fluconazole bound to CYP51 from T. cruzi. It was shown that at least posaconazole and ravuconazole are highly efficient against Trypanosoma cruzi, and ravuconazole is getting into clinical trials with Chagas disease patients. However, there are issues related to intellectual property, price, toxicity, formulation, and drug resistance with these antifungal azoles. And therefore, people like we in academic environment work on development of new chemical scaffolds to make more drugs against Chagas disease. And in our group, we use structure-aided drug design based on high-throughput screening hits. And currently, we have two different directions. First, this is hit-to-lead optimization of previously defined non-azole compound, which we call LP10, and de novo screening of SMDC library in a search for new scaffolds, and exactly this part of work was funded by CEND Center last year. And I will give you a brief update on the first direction. LP10 hit was derived from high-throughput screening efforts on Mycobacterium tuberculosis CYP51, like about 10 years ago. Through structural characterization and some rational drug design, we came out with the compound which binds Trypanosoma cruzi CYP51 very tightly. And as I already demonstrated, it is quite efficient in destroying cell membranes in unicellular organism, and it also showed very good efficacy in mouse model of acute Chagas disease. Therefore, we started to do heat-to-lead optimization, and during the last year, we determined which specific stereoisomer is active. So it turned out that this one has the same nanomolar affinity as the racemic mix, while the second stereoisomer has 2 orders of magnitude weaker binding. And we also work on co-crystallization with CYP51, trying to obtain crystals, which we are obtaining. And this will follow up by solving structure of this complex. Meantime, in collaboration with Scripps, Florida, we do some optimization of compound in terms of stability in the liver microsome assay. Our goal here for this assay is to get 30 minutes lifetime, because currently our compounds are short-living compounds here, as you can see. And we already synthesized in Scripps next generation of compounds which we are testing for binding affinity. Some of them retain very high binding affinity to the target, and we will be testing them for drug metabolism pharmacokinetics as well. So now going back to the second part of our research, de novo screening. So we choose to use physical properties of cytochrome P450 to screen the library against our protein. And this physical properties is defined by the presence of the cofactor heme, which is here in the substrate binding tunnel. And when we have inhibitor binding in the active site, which is capable to form coordination bond to heme iron, we get this type of spectrum, which we call type 2, meaning that coordination bond to the iron is formed. Or if we have compound of the substrate type, which bind in the active site but does not form coordination to the heme iron, we obtain different type of spectra, which we call type 1. And of course, coordination bond is this strongest single interaction in this complex. And we can assume that compounds which can coordinate to the heme iron will be stronger inhibitors. So we basically focused on the search of type 2 inhibitors. The advantage of this assay is that we detect only those interactions which are in the active site. We do not need any redox partners to drive these reactions, and we actually do not need to know the catalytic reaction which this protein performs, which sometimes is adventurous for some other cytochromes P450s. However, assay has downside, which is limited sensitivity because of UV visible spectroscopy is used for detection, and slow reading if we choose to use spectral mode. This is brief design of the assay we used. So to test one set of compounds, we need to read 2 plates, because one plate, they are identical with an exception that one plate contains protein and another one contains only compounds. We have 2 lines here, 2 columns for correction, for background correction. We use this signal from here for background correction, and the last line usually has reference compound. So after all these wells are corrected for background, we subtract plate A from plate B. And ideally, if there is interaction between target protein and compound in the well, we expect to obtain this signal if we detect a response in the spectral mode, which is from 350 to 500 nanometers, where Sareb band of iron shifts. However, it's clearly this more rigorous assay when we detect in the spectral mode. However, it's understandable this is much slower process. And alternative here is to read only 2 wavelengths, maximum and minimum, and then subtract it from each other and follow this difference as a monitor this difference as a screening signal. And due to the time issues, we opted to run 2 wavelength screening, and we proved that it's good conditions to use when protein concentration is 2 micromolar, and we choose to use compound concentration at 12 micromolar. After all screening was performed for 100,000 compounds in a 2 wavelengths mode, we identified 2,700,000 hits, which satisfy this requirement. The difference is in absorbance is higher than that and lower than that. And this constitutes 2.6% of the entire library. To validate how many of those hits are false positives, how many are false negatives, we choose to select 80 compounds and screen them in the spectral mode with serial dilutions and cross-validate these same 80 compounds in the whole cell T-Cruci assay. So to choose 80 compounds, we roughly divided the 2,700 hits in 40 structural groups and pick up 2 compounds from each structural groups. This is how we acquired those 80 compounds. And criteria to pick them up were as following. Absorbance has to be higher than this number, but lower than that, because this is maximum when 100% of sites are saturated. And we also choose to pick up compounds from the outliers and see if we find any positive hits in this group of compounds. So these 2 slides simply demonstrate you every single compound which was chosen to run validation and cross-validation in biological screen. And again, to remind you, for positive hits, we're now looking for this type of curve, and we use now 4 different concentrations of compounds, running from 12, which originally was used for screening, going up to 2 micromolar, which constitute 1-to-1 molar ratio, and LP10 is used as a reference, and this is a saturation curve at 1-to-1 ratio for LP10. And needless to say that since we expect compounds with high affinity, we are looking for those which do not have any difference in the curve, where this amplitude of the curve is independent of concentration. This would mean that we found a really tight binder. For false positive hits, we expect to have something like this: no expressed maximum and minimum, and 2 curves run approximately, 2 flat curves running approximately parallel. What we found is that 80% of our compound we selected for validation are false positives. Less than 20%, which constitutes 15 compounds, are true positives. And this is how their curves look like. But with exception of 1 single compound, which is shown here, all curves show concentration-dependent variation of amplitude. Only single compound, which is shown here, gives us ideal pattern, what we are looking for here. And in parallel, all 80 compounds were validated in whole cell T-Cruci assay, which is based on fluorescent dye. This is a fluorescent image of a mastigot of mammalian cell infected with T-Cruci mastigot. What we stain here large is mammalian cell nuclei, and small one kinetoplasts of trypanosoma. So this way we can easily count how many T. cruzi cells are present within one mammalian cell. And this assay is low throughput screening, automated low throughput screening. So we passed all 80 compounds through this assay, and we found that 5 compounds out of 80 have inhibitory effect in Trypanosoma cruzi. As a reference, we used the only clinically approved Chagas disease drug, benznidazole, and these are numbers for benznidazole. The only compound from these hits which could reach 100% of inhibition was this compound in the upper right corner. And interestingly, that 100% inhibition was achieved at concentration 4 times lower than concentration of benznidazole. Now our question was how these two sets of compounds overlap. And we were very pleased to see that biologically active hits entirely overlap with 5 truly positive hits, and compound which was best in the biological screen was also best in our screening assay. So now we can make conclusion that 2 waves mode is more efficient time-wise, but we generate too many false positive hits. However, on a positive note, structurally diverse true positives are also in this pool. And even more pleasant than that, true positives correlate well with biologically active hits. What we are going to do now? So since we validated only 80 compounds, we have more than 2,000 left, about 2,000 left. However, this time we decided to analyze structurally what is similar between those positive hits. And sure enough, we saw that all of them have aromatic nitrogen in the position capable to coordinate to the heme iron. And when we sorted out the library of 100,000 compounds based on the structural features, we identified more than 6,000 compounds which have such features. However, around 2,700 of them were silent in the initial 2-wavelength screening, and 3,300 gave us some kind of a signal, and 1,300 gave a signal which we would consider as a signal of hit. So now we have about 1,300 compounds which we would like to rescreen in the spectral mode with compound concentration about 2 micromolar in 1-to-1 molar ratio. And right now we're strategizing with SMDC how this will be done, whether we are going to go after all 1,300 or we are going to dissect this further based on structural similarities and opt for screening, let's say, 500 or 400 compounds. So this, we are working on this strategy, and we will come up soon with the decision. And naturally, our next step would be to validate those 1,300 compounds and cross-validate those hits in for biological activity. And of course, as structural biology lab, we would like to take already validated hits into structural studies to structurally characterize complexes between inhibitor and CYP51 and do hit-to-lead optimization. And here I would like to finish and acknowledge people who were involved in this project. Of course, this is my group represented by 3 people here. Shamila Gunatileki is present at this meeting, and I guess you can meet her during the coffee break. Other people from the CEND Center involved in this work were James Makiro, Juan Angel, Jerry Good and Patricia Dole. And of course, we collaborated in a very, we spent lots of time discussing project and strategies with SMDC. They were very helpful, particularly Kenny and Joe. They helped us to sort out compounds and choose compounds for cherry picking. Scripps Research Institute, Bill Rausch Laboratory, collaborate with us on hit-to-lead optimization of LP10 compound. And of course, we're always grateful to our beamline, represented by James Holton. They help us immensely all the time we need help. And the money for this research came mainly from three sources: Sandler Family Foundation, CEND at UC Berkeley, and NIH. And thank you all of you for listening. This is an interesting piece of information. Our work was somehow covered on the UCSF website with this article here, which announced that we received the CEND award and described a little bit the content of our work. This was read somehow with the representative of Russian consulate in San Francisco. They decided that this award is somehow equal to Nobel Prize and, and decided to make a reception at the consulate where some colleagues of mine, like James McCarroll, gave a speech about the importance of research on the neglected diseases. And of course, I was honored as an award recipient. So thank you very much, CEND. So questions, of course.
Yes. Questions? Do we have a microphone? For now, just project. Okay.
How potent was that lead that you found from the full suite? The potency of the hit.
So potency, at least we got, we did it only in the culture assay against parasite in the culture. And I would say like 4 times more potent than benznidazole, which is the only single clinically approved drug for Chagas disease nowadays.
So it's as potent as LP10, or wasn't clear?
We did not make this comparison yet because this is our latest finding. We did not really fully characterize our compounds yet. So this I cannot, but in the assay which we ran, it was 4 times more potent than the reference, and the reference was benznidazole.
So I wanted to ask if that was the appropriate reference, because you started the talk by saying one of the reasons that more drugs are needed is that the potency is low and the side effects are high, and certainly those two things are related. So I guess what I'm really asking you is what level of potency would you want to achieve to get actually a better compound than what's currently available?
Well, we certainly will be working on that, and we will address the potency versus like very potent inhibitor posaconazole and LP10. We just at the beginning of this work. However, you are right, benznidazole is not very good comparison because it's quite an efficient drug. However, this is a drug which accepted for treatment and somehow standardized these assays. People run screening versus like having benznidazole as a reference, something which is yeah.
Just to follow up on Gideon's question, did you measure the binding affinity? So you know it saturates at 2 micromolar, but the LP10 had a nanomolar affinity.
Mm-hmm.
How did the compound that came out of the library just make it straight out of the library? How did that compare?
Oh, no, LP10 didn't come straight out of the library.
Sorry.
LP10 was next generation of top hit which was found in the library, and we added this imidazole tryptophan ring to this molecule to make it as potent as it is.
Right. So I'm asking, how did the compound that came out of the library compare to that in affinity? Just so that
we did not run this yet.
We get an example of how much optimization you might have to do to reach the potency of LP10.
Right. These experiments are not yet done. The manual part of this work is not done yet. So this is what we got from screening. And essentially, we got one compound which was taken into manual investigation. So this will be just our next step to do.
But of course, you know, Tom, that the affinity for the target is one of a list of 20 things, right? So benzodiazole treated you to treat for 120 days, is that right? So there's a lot of room to improve in the target therapeutic profile. If your drug of choice, you need to treat for 120 days, as you in tuberculosis know well. You're like, only 120 days? What are you complaining about?
Okay. Let me just close my slide.
Okay. We're going to slowly introduce the contestants now while he loads up. So Jonathan Gable is in...
Can you speak into the mic?
Yes. Is this one on? No. Okay, thanks. I'm getting the thumbs up there. So now it's my pleasure to introduce Jonathan Gables from Charlie Craik's lab at UCSF. He's going to talk about Kaposi-sarcoma-associated herpes virus and an interesting serine protease that's critical to the virus, but as you might know, serine proteases are very difficult to inhibit by traditional drug discovery, and so the approach that they've taken is looking for things that interrupt the dimer. So the enzyme needs to be a dimer to be active. Okay. And that was a little teaser. Okay, do you want to put on the lapel mic, or do you want to?
Is that going to be fine in terms of volume? So as was stated, today I'm going to discuss targeting a herpes virus protease as a means of combating Kaposi sarcoma. So Kaposi sarcoma is a lethal cancer that disproportionately affects HIV/AIDS patients in sub-Saharan Africa. For instance, in Zimbabwe, KS/AIDS patients suffer from rapid disease progression, as well as life expectancy is often less than 6 months. And in Uganda, by age 11, 50% of children have been infected with the etiological agent of Kaposi sarcoma. So KS is ultimately caused by a viral infection, and that's infection with Kaposi sarcoma-associated herpes virus, which under normal circumstances, the immune system will more or less take care of. However, in the immunocompromised population, KSHV infection results in KS. And so as such, as the prevalence in HIV/AIDS increases and as resistance to HIV/AIDS treatments increase, we see a concurrent increase in the severity and prevalence of Kaposi sarcoma. To date, there are no KSHV-specific treatments, and current treatments focus primarily on treating HIV/AIDS. And so our goal then is to find a small molecule that can limit KSHV infection. And inspired by recent successes in inhibition of viral proteases, we decided to target KSHV protease. So let me provide some biological context for this. So KSHV protease is expressed as a monomeric zymogen fused to the viral assembly protein. And its expression occurs in the cytosol along with the major capsid protein. These proteins associate and are ultimately transported into the nucleus where the immature capsid forms. As this formation occurs, there's a concentration-dependent dimerization of the protease that results ultimately in release of the protease from the assembly protein and activation through dimerization. This active protease can then go on to cleave the assembly protein, resulting in capsid maturation and ultimately in infectious virions. And so our goal then is to target this protease, eliminate its activity, thus disallowing the maturation of the capsid with the goal of preventing KSHV infection. And so we have a problem and we have a goal, and the question is, what tools do we have available to take on this challenge? And in the lab, we've developed the biochemistry and molecular biology necessary to produce reagent quantities of KSHV protease. We've then taken that KSHV protease and developed a high-throughput screening assay based on a fluorescent peptide substrate. We can then take hits from this screening, immediately pipeline it into a medium throughput NMR and crystallographic studies to get at the mechanism as well as mode of inhibition. Promising compounds that come out of this can then be put into cell culture assays to test for ex vivo efficacy. And so we've implemented all of these strategies on a small scale and arrived at a molecule that just simply isn't pharmacologically viable. But I want to tell you the story of where we've gone and where we ultimately want to go. And that starts with a little bit of history. So it turns out that, perhaps not surprisingly, we're not the first people to think that we could inhibit this herpes virus proteases. And so drug companies have attempted this, but the focus has always been on the active site. And through crystallographic studies in the lab, we were able to show that, in fact, the active site is quite shallow and solvent exposed. We believe that this is the reason why these screening efforts yielded only covalent inhibitors and molecules with relatively poor pharmacological qualities. Ultimately, for these reasons, KSHV protease and herpes virus proteases in general were deemed undruggable. But by knowing the biochemistry of this molecule, of this protease, we were able to see that there's an alternative weakness that we can exploit. And that weakness is namely that the protein is only active as this weak dimer and inactive in its monomeric state. And so what we envisioned was generating a small molecule that could shift this equilibrium away from the active dimer to the inactive monomer. But ultimately, in order to find such a molecule, we needed an assay. And so this is when we developed a fluorogenic screening substrate where the cleavage at the cis-aryl bond results in release of coumarin and an increase in fluorescence. We then, in collaboration with Kip Guy at St. Jude Children's Research Hospital, were able to take this assay into a small-scale, high-throughput screening context, screen a 180-member alpha helical mimetic library, which ultimately resulted in a single hit with an IC50 of about 3 micromolar. The molecule, with slight modification for solubility reasons, is this, DD2. We were able to then take that into our NMR screen, where we can monitor the monomer-dimer equilibrium via selectively labeled methionine at the dimer interface, and show that with the titration of our molecule, we get shift to the monomer population. We could then verify that our NMR screen is functioning using simple size exclusion chromatography, where in blue is the control DMSO. There's a clear dimer-monomer equilibrium. With the addition of inhibitor, only the monomer peak remains. Ultimately, we were then able to take DD2 and KSHV and co-crystallize it, resulting in the definition of a specific binding pocket at the dimer interface, thus allowing for virtual screening efforts as well as more structure-based design. While we were really excited about this process and the progress, we ultimately realized that this DD2 molecule's drug viability or pharmacological viability is fairly limited. And so as such, we know that we need to find a better scaffold, a better molecule. And so we took our fluorogenic substrate, and again, with the help of Kip Guy, we're able to generate a true high-throughput screen with a z-score of better than 0.7 in a 384 well plate format, optimized for these key parameters, and validated in a 4,000 compound screen with our DD2 molecule as a positive control. Then to further reduce the potential of false positives, we also developed a secondary IFRET screen using a different substrate, different excitation emission profiles that allows us to reduce the false positive rate. We can take molecules that succeed in being funneled through all of these processes, and with the help of Don Ganem at UCSF, put this into a cell culture screen where we can monitor viral latency based on GFP expression, viral lytic cycle based on RFP expression, take the spent media from this lytic cycle, reinfect with healthy cells, and then monitor the extent of reinfection, where we would expect a protease inhibitor to reduce the ability of the virus to generate new capsids, thus limiting the viral titer of the spent media and ultimately resulting in a reduced viral infectivity or reinfection. To summarize, let me take you through sort of our grand vision for what the project is. We can take a library of compounds and put it through our fluorescence-based KSH3 protease high-throughput screen. Molecules coming out of this screen can be immediately put into a medium-throughput NMR and crystallographic studies to get at the small molecule binding mode and mechanisms of inhibition. Promising hits from that process can be taken into cell culture to test for ex vivo efficacy, ultimately, hopefully, resulting in a KSH3 protease lead inhibitor. And what makes this project additionally exciting for us is that because of the structural and functional homology between all 8 human herpes virus proteases, we believe that a KSHV protease lead inhibitor may very well also be an inhibitor of these other HHV proteases, as is in fact the case based on our preliminary data for DD2, where we've shown that DD2 will inhibit members of all 3 subfamilies of herpes virus proteases. And so we've gone through this process and shown proof of concept that we have the tools necessary to arrive at a small molecule, but our small molecule simply isn't pharmacologically viable. And so what we need to do then is to expand our search in chemical space to arrive at a more reasonable molecular scaffold. With that, I'd like to thank Charles Craik, my advisor, Greg, a postdoc in the lab who was integral in setting up the NMR experiments, Ida, our resident crystallography expert and postdoc in the lab, Tina, a fellow graduate student in the Craik lab who's been instrumental in doing a lot of the molecular biology and biochemistry. Also, our collaborators, Kip Guy at St. Jude Children's Research Hospital, who helped really facilitate the creation of our high-throughput screen. Don Ganem for all of his assistance with the cell culture work, as well as Tom Meek for providing some of the plasmids for the expression of these proteases. Last but not least, of course, I'd like to thank the CEND organizers for putting together this great event and giving me the opportunity to speak, NIH for funding, and ultimately you for your attention. At this time, I'm happy to take questions.
From the judges.
The false positive rate. So how high was that false positive rate to begin with?
I don't actually know what the false positive rate was for the 4,000 compound screen. We only had 3 hits that came out of that screen. I believe one of them was an aggregator, one of them was a detergent-like molecule, but just obviously looking at them. And then the other was toxic. So 4,000 molecules was not enough.
So you mentioned that you don't like the pharmacological properties of your lead. So I'm a medicinal chemist. I wasn't that put off by it. What do you see as being wrong with it, and what are you looking for in your next generation?
So in its current state, probably the biggest problem is just solubility. So right now it's soluble up to maybe 80 micromolar tops with an IC50 of only 3 micromolar, and that's with some DSMO in solution. So solubility is a #1 problem. I would say that's actually the #1 problem. There's also the potential for limited ability to extend off of that scaffold from a chemical perspective. So what we would look for is a more soluble molecule that can be further derivatized to take advantage of potential other grooves in the protein, for instance.
The high throughput screen you set up was probably not so different from screens that were used in the past. So do you think that in the past screens, a dimer disruptor they would have discarded for some reason?
That's something we've always wondered about. And we think that it's probably on account of maybe the solubility, possibly that they just didn't screen alpha helical mimetics, but one would imagine that those would have been somewhere in their library. But through talking to people who have done that, such as Tom Meek, it seems that for whatever reason, it was missed.
And the other question I had is when you solve the crystal structure of DD2 with the, is it a dimer in the structure, or is that?
It's an asymmetric dimer, but only due to crystallographic contacts. So actually, we truncated, go back here. So these, it's hard to see with the lights, but these two helices are unstructured in the inactive monomer, and that allosterically causes loss of activity at the catalytic site. And so in order to crystallize it, we've truncated off these two floppy helices. And we've shown through NMR that truncation of these helices doesn't affect the KD of binding of our small molecule. But so there was an asymmetric dimer, but it was crystal contacts only, because with this truncation, it's an obligate monomer. It won't dimerize.
So before doing a bigger screen, have you thought about doing rational drug design, like efforts to maybe do some virtual screening before you do actual screening to get more hits?
Absolutely. I just started virtual screening on this molecule. The crystal structure is, as of yet, unpublished, so we just generated that. But absolutely, we're going to, just some of the virtual screening hits suggest that we can not only target this really hydrophobic pocket, but that we can extend out into a more hydrophilic groove to get some orientation and some hydrogen bond formation that could aid in structure-based drug design.
Okay, I have a couple questions, one technical and one philosophical. So the more philosophical question is, if the binding interface, the protein-protein interface is 1 micromolar, what do you think that says about the inherent binding affinity of that site? Do you think that you're going to be limited in small molecules just because there's not that much there?
Right, so there's a lot of hydrophobic surface, which I think we can exploit. But to gain specificity, we'll need to, that's what I was talking about branching out sort of away from that particular pocket and binding then to some hydrophobic or more hydrophilic portions of the protein.
So outside of the interface altogether. So actually a molecule that's bigger than the protein-protein interface in a sense? Or
only one that binds in that pocket and then wraps around. So actual surface area will probably be much less than the protein-protein interface as a whole, but it wraps out away from the interface and into a more hydrophilic environment.
Mm-hmm. Okay. Then the technical question was, the FRET assay is probably going to have, maybe have fewer artifacts than a fluorogenic cleavage assay. Does it have enough signal to background to run that as a primary screen?
I was not actually the one who developed that, but I don't really have a good answer for that, but it would be something to investigate, certainly.
Check out, yeah.
I have another also philosophical question more about the Kaposi sarcoma and neglected disease aspect. So can you elaborate a little bit more on that? You know, what the benefit would be of a dedicated Kaposi sarcoma drug versus making HART more available to the patient population, which would also help against Kaposi sarcoma?
Sure. Yeah, so I mean, if HART was completely readily available and there was no resistance to HIV/AIDS treatments, that would be the way to go. No doubt about it. But in light of the fact that there are resistance instances, and that in sub-Saharan Africa, HAART just frankly isn't available to the whole population, there is an unmet need for this type of Kaposi sarcoma specific treatment.
Do you think your drug is going to be available?
It would have to be a small molecule that was, a single treatment that really could be produced in large quantities cheaply. Absolutely. Thank you.
Thank you very much. Yeah, you don't want to be caught on mic saying something mean about him. Andrew, you want the phone? Sure. Okay. Now, while he's setting up, I'm going to introduce Andrew Maley from Doudna's lab, Dr. Doudna's lab at Berkeley, and his talk is entitled "Catching the Flu," about looking at ways to treat influenza virus using a different kind of cell-based assay, image-based cell-based assay.
So I want to thank the organizers for this great opportunity and just kind of start out with this little picture here, which I think really emphasizes the current state of the art, where we're each year being kind of combated by different strains of influenza virus, and we're a highly susceptible population. But this is all the protection that we have, and I'll tell you a little bit why that might be the case and our plans to expand our coverage using antivirals. And as we're all aware, starting last year, there was a new pandemic virus that emerged in Mexico. This was the 2009 H1N1 virus. This virus rapidly was transmitted from Mexico to the United States, and then around the world in just a few weeks, where it was declared a global pandemic. Since this pandemic was over, and that only happened in August of 2010, there were between 40 and 90 million people that were infected by this virus. And while it was highly transmissible, we were lucky in that the pathogenicity was quite low, yielding about 9,000 to 18,000 deaths. And this is on par with seasonal influenza virus, if not a little bit lower. But then on the extreme opposite end is bird flu, or H5N1 influenza. And since its outbreak in 1997, there have only been 500-odd confirmed infections in humans. But this is a virus you don't want to get. There's a 60% mortality rate. And what keeps influenza epidemiologists up at night is the idea that you could get a highly transmissible virus such as this one that acquires just a little bit of the pathogenicity of that virus. And that's thought to have occurred in 1918. This was the mother of all pandemics, the Spanish flu. And this occurred at a time where many different things came together to basically result in a pandemic that killed about 50 million people worldwide. And it was predicted by a paper at the CDC that if something like this were to happen now, we'd have similar rates of mortality, but also it would cost between $70 and $160 billion. Now, despite this large exposure to pandemics, we also have seasonal influenza, and it's easy to forget that garden variety influenza kills between 20 and 36,000 people in the United States every year alone. So influenza is a RNA virus. It contains a negative sense RNA genome, and as such, it has its own viral polymerase, shown here as PB1, PB2, and PA. In the absence of this polymerase, the virus is dead in the water, and in fact, it's negative stranded RNA, so one of the very first steps that has to happen in an infection is you get transcription by the viral polymerase. Now, I mentioned there were limited treatment options, and in fact, there are only 4 compounds available for treatment in 2 different classes. We have the adamantanes that target the influenza M2 ion channel, and these are amantadine and romantadine. And we also have neuraminidase inhibitors, oseltamivir and zanamivir, known as Tamiflu and Relenza. And in fact, this is from the Bay Area, Gilead. Now, during influenza infection, you get attachment at the cell surface and entry, where then the virus deposits its genome into the cytoplasm that transits to the nucleus. And this is where all the fun takes place. The viral RNA-dependent RNA polymerase mediates replication and transcription to produce new genomic RNAs and viral proteins that then assemble at the membrane surface and bud off. The 2 compound classes that are out there block different stages of infection. Amantadine and romantadine prevent acidification of the core of the virus so it can't deposit its RNA into the cytoplasm, whereas neuraminidase block this late-stage event where viruses can't be released from the cells. And while these compounds are effective, they have limited utility, and that the virus has rapidly evolved resistance. So there are many different primary isolates circulating that have resistance to M2 inhibitors by simple point mutations. And then even within the short time that the 2009 virus has been around, resistance to the neuraminidase inhibitor Tamiflu has evolved. And normally resistant viruses take a hit in their fitness. But what influenza has been able to do is evolve second site suppressors so that viruses that are resistant to these drugs still show wild-type fitness. And this really screams out for yet other treatment options and different targets that we can use to try to prevent or treat influenza infections. And at the heart of this entire process is the RNA-dependent RNA polymerase. Influenza's polymerase is a 250 kilodalton trimer composed of the subunits PB1, PB2, and PA. PB1 is the structural and the catalytic core. It actually has the catalytic activity of the polymerase. And it plays a role in both transcription and replication of the viral genome. PB2 and PA have best defined roles in a unique transcription event called cap snatching. So this is a very exciting and interesting process that influenza uses, where it binds the host messages by their mRNA at the 7-methyl G5' cap. These messages then are cleaved by an endonucleolytic site in PA to produce short capped oligos. Those capped oligos then are used by the PB1 subunit to prime synthesis of viral messages that are both capped and polyadenylated. In addition to the polymerase, you need the viral nucleoprotein. This is an essential cofactor that binds to the polymerase and the viral RNA, and it's thought to chaperone the assembly of the ribonucleoprotein complexes that do all of these functions. So to study the influenza polymerase and establish a system by which we could identify compounds that would inhibit its function, we created a cell-based activity assay. And in these assays, we can express the polymerase along with nucleoprotein in cells, and then also express a viral reporter here, which is effectively just a viral gene that now encodes our favorite reporter, which is GFP. Only in the presence of a polymerase does this reporter get replicated and transcribed. It's in a minus sense, so when the polymerase is absent, the cell doesn't know what to do with this RNA. When the polymerase is present, we then get GFP that we can visualize and quantitate. And as a proof of principle experiment, I show right here some fairly striking results that in the presence of a wild-type polymerase, we see a high degree of activity, whereas in this mutant, which is an attenuated form of the polymerase, we have very low, if no activity at all. We can quantitate these by FACS analysis and see that there's about a 5-fold difference in the activity between our wild-type and mutant forms, and that this represents a 1 to 2 log difference over those that are lacking polymerase subunits to begin with. And this difference becomes greater as the assay goes on, because the polymerase not only just transcribes, but actually amplifies the amount of template that's there for its function. I've now used this assay for over 2 years, and we see that it's a very easy assay that I've gotten undergraduates to do. It has a large dynamic range, which is up to 4 logs of dynamic range with GFP and over 6 logs when we use luciferase. It's highly reproducible from well to well and from day to day. And unlike assays that are done using actual virus infections, this assay focuses only on the RNA-dependent RNA polymerase activity. And when we use a 96-well format with luciferase, we had high z-scores over 0.8. What I would envision, and this would be done in collaboration with the Small Molecule Discovery Center, is a pipeline where we would have cells that are expressing the polymerase protein's nucleoprotein in our reporter. We would then use high content analysis to identify cells that were targeted by active compounds and prevented polymerase function, comparing those to that are inactive or our control population. That data then would be analyzed with a center where we would enrich for compounds that have FDA approval. We would eliminate compounds with known effects on toxicity, and then cherry pick these for further analysis. These compounds would be validated in 3 different ways. First, we would switch over to a luciferase assay to eliminate all those compounds that simply target GFP. And this is a much more quantitative assay, so we could rapidly determine things like IC50s. We could then counter-screen against RNA-dependent RNA polymerases from other viruses, such as vesicular stomatitis virus, arenavirus, and bunyaviruses. These use different strategies for their replication, and in fact, these 2 are also cap-snatching and represent emerging diseases such as Bunyamwera virus, Hantan virus, Lacrosse virus, and Lassa virus. And then finally, we would test the effects of these in infectivity assays. We have culture-based assays where we can measure virus replication kinetics in the absence or presence of compounds. And we have animal models with mice infections that allow us to test the effect of these actually in an animal. From these, we could then use biochemistry to determine the mode of action. We have a handful of assays that I've developed in the lab that allow us to look at discrete functions of the polymerase. Primer extensions will allow us to tell whether we've got defects in transcription, replication, or both of those processes, and that will allow us to narrow down the potential targets of the drug. We have assembly assays that will allow us to look at whether or not the influenza trimer is assembling and whether that trimer then forms the ribonucleoprotein particles that are necessary for function. Others have established endonuclease assays with a purified component of PA that allow us to test its function. And then I've established in vitro assays using purified polymerase where we can measure the cap snatching and extension that requires PB2 and PA, and then the primer-independent replication that primarily relies on PB1. And luckily for us, there are structures of certain domains of the polymerase complex. So the structure of the endonuclease domain from PA, and it appears to be a classic 2-metal active site. And this creates a nice large pocket that you can envision is, might be targeted by these compounds. So if we were able to demonstrate from our biochemistry that the PA subunit's being targeted, we could attempt to perform crystallography in the presence of the compound. And that would allow for future refinement. Similarly, the PB2 cap binding domain has also been solved. And here we can see the 7-methyl-G cap is bound by this aromatic sandwich. And it too presents a nice pocket that you can envision compounds sticking to. And we could use crystallography once again to drive the refinement of these compounds as well. So what I've told you today is that we have a validated cell-based assay that we've been using for over 2 years now that's highly reproducible and it's amenable for high throughput screening and high content analysis. We have a large number of reagents in the lab to both test the specific activity of these compounds against the polymerase and identify their mode of action, while also asking the question, are these compounds functioning against other cap-snatching viruses, including those of emerging diseases like Hantan virus? We've got in vivo assays to determine the efficacy of the compound and biochemical assays that will give us a mode of action, and there are some structural options that allow us to refine these compounds as we go forward. And ultimately, I think what I've also tried to show today is that there's a strong need for these improved therapeutic options, not just relying on the 2 that we have. And this isn't just for pandemic viruses, but also for the standard influenza that we face each year. And so now we come back to our little man in the rain. And what I'd hope to do at the end of this screen is be able to take this little shield we have and expand it out here, give us many more options for treatment and prevention, because these viruses themselves are not going to go away. This work was all done in the lab with Jennifer Dowden and with great suggestions and advice from both Jennifer and the lab members. And I owe thanks to our funding from the NIH. And I'd be more than happy to take any questions. Right, so since we're using high content analysis, one thing that we can do is also co-transfect in something like RFP, just to make sure that the compounds that we're working with aren't simply, like you're saying, disrupting protein synthesis, or the reporter requires Pol1, so maybe it's a defect there. And if we have RFP, we can also quickly look at toxicity and just simply count how many cells are there and transfected.
So I have another question. What makes you think that this target is a lot better than the other targets that have already been exploited pharmacologically? Is it more conserved between viruses? Or?
Right, so each year there's a new version of flu that comes out. And from year to year, those differ on the amino acid level by maybe 1%. But within the polymerase, there are absolutely conserved motifs that just never change. And in fact, some of those are the classic RNA-dependent RNA polymerase motifs that you see, this DDE motif. Within the endonuclease domain, there's not much variability. So the other reason that I think this is a good target, in addition to its conservation, is that it's got multiple activities, all of which, when impaired, will disrupt function. So if you can block protein-protein interactions, the polymerase doesn't function. If you block the cap binding or the endonuclease or the polymerase or the RNA binding sites, it doesn't function. So in addition to just targeting one drug to one particular active site, what we would do with this screen is we'd screen at least 5 discrete possibilities.
Just you know, one of the reasons that we don't that cell-based screens are often not run is you will require cell permeability, which is of course something you will need eventually. Yeah. But I guess separately from that, so you're assembling this complex and the substrate in the 293T cell.
Yes.
Do you have some validation that represents, with perhaps pre-existing inhibitors, that represents the same enzyme that's actually found in normal
Right, so
process?
it's a good question. Can we honestly and faithfully recapitulate what the polymerase does in an infection setting? And we have biochemistry that says that we can do that. So we can look at the kinetics and the production of discrete species of RNA when we transfect and compare those to the similar things that happen in a cell. And what we do see is we see that the majority of the RNAs that are made in our transfected system mirror those that are found in an infection setting. There are two compounds that have come out of various systems that seem to target the polymerase. One is a general RNA-dependent RNA polymerase inhibitor. It inhibits these type of cell-based assays and similarly inhibits the viral infection. The other seems to be something that prevents the polymerase from assembling with nucleoprotein, and its best guess is that it aggregates nucleoprotein. And when those came out of screens that were done on an infection setting, they could then take them into an activity assay and see a similar result. And to get back to the first point, certainly there's a lot more complexity that occurs when you do a cell-based screen. Unfortunately, at this point in time, it's not realistically possible to get enough recombinant polymerase to do a high-throughput screen in that fashion. So I think this is the best we have at the time.
So a technical question, which might be naive, but there you have it, and then a philosophical question. So the technical question is, you mentioned that there's an amplification effect, right, which basically changes the signal over time. Yeah. How do you account for that in your assay, and how do you plan to account for it when you start screening? How many compounds do you have, Michelle? Yeah, it's going to take a while.
Yeah, so what happens is that as you go forward, you get a shift from message production to virus replication, and then you get this wonderful, I mean, it's pretty amazing. These cells will be green in front of your eye, not under the microscope. And we're faced with this problem right now because we're doing some shRNA screening as well. So in those cases, what we do when we want to stop that, we simply fix the cells or we treat them with puromycin or cyclohexamide or something like that. That's the best case we have right now.
Yeah. Okay, so the philosophical question is, you mentioned you've got a multiplicity of targets, which is a great feature of an assay like this. Not all of them are going to be good.
Right.
If you were a betting man, and I'm not saying you are, what do you think the best one is and why?
Well, I think the best one is also the hardest one, and that's going to be the actual catalytic core of the polymerase. And the reason I say it's the hardest is we have the least information about it from a structural standpoint. It's just been the part of the protein that's not been amenable to anything. The reason I think it's the best is that it's so different than what a cell does. There's no reason for the cell to have an RNA-dependent RNA polymerase. And when you look at things like some of the initial drugs coming out of HIV, what did they target? They ended up targeting the reverse transcriptase. So we could envision nucleotide analogs or something like that popping out of this screen. And we know from past that those are druggable and amenable. It's not to say that we won't find something else or won't find something that's better, but that's where I would lay my money.
Any other questions from the judges or the peer judges, the peanut gallery?
Thanks.
Okay, good. While the judges are deliberating, I'm just going to give you a brief introduction to the Small Molecule Discovery Center, to some of the neglected disease targets that we've worked on, just a smattering, just to give you an idea of what some of the capabilities are, and to encourage you, if you have any projects in high-throughput screening, lead optimization, technology development, to feel free to come talk to us. We do have a core facility. The high throughput screening facility is a core facility, so open to anybody. And the rest of the work we do is collaborative, so usually through writing grants together or other sources of funding. Okay, so the Small Molecule Discovery Center is basically involved in doing small molecule discovery, lead discovery, in an academic setting. So let's talk about some of the joys and challenges of trying to do that, especially for neglected diseases. So the mission of the SMDC is to advance our understanding of cell protein function. So to develop compounds that can act as chemical biology tools to help us understand all of this novel biology that you've been hearing about today. Through that work, we'll also hope to identify promising new drug targets and small molecules that can serve as lead matter for those targets. And then also to advance our technologies for our so-called challenging targets. So a particular interest of mine is in protein-protein interactions, one of which you heard about today. Some of the particular molecular recognition and structural challenges with those types of targets will probably require novel technologies in many cases. Okay, so the Small Molecule Discovery Center has expertise in this range of the drug discovery pipeline. So target ID, we don't usually do that. That usually comes from our collaborators, like the Sandler Center for Drug Discovery. We work with people in hit identification, hit to lead, lead optimization, and then we have some expertise to help people partner with safety and toxicology to do IND enabling studies. And then some of us have worked in clinical development in the past, but we pass that off to experts at the NIH or in pharmaceutical companies. Okay, so the tools that the Small Molecule Discovery Center has for doing some of these things include the screening core. We have 180,000 molecules. We routinely screen about 150,000 of those. We also have a human siRNA library. We work both in cell-based assays, high-content imaging assays in particular, and biochemical assays. We have our own informatics system. Informatics is a really important aspect of getting the most out of your high-throughput screening and lead discovery data. It gets to be very complicated very quickly. And we do about, we actually did 23 high-throughput screens last year. Now, supporting that effort is our interest in screening technology. So one of the technologies that we focus on for challenging targets is fragment-based drug discovery, which I have a slide about if I get to it in time. We also do a lot of high-content imaging. I'll talk a little bit more about this project. This is, these are schistosomules. They're baby worms and blood flukes. And they're a very challenging target for high-content screening and other challenging targets like protein-protein interactions, allosteric interactions, challenging organisms. And then after you have material from fragment screening or high-throughput screening, there's a lot of chemistry that goes into turning those into really good, selective, potent tools and drug leads. And so we have a lead discovery group led by Adam Renslow. He's a medicinal chemist. We also support some of the biology. And we have large collaborations focused in parasitic diseases, as we'll talk about today, also in neurodegenerative diseases, and some in cancer. So basically, we'll work on anything, but this is what people, this is what academics have largely been focusing on: neglected diseases, orphan diseases. So a neglected disease, we could say, is something that lots and lots of poor people get, or that a very small number of people get locally, so that it's not, with all the big problems that you have to tackle in drug discovery, these are not the ones that rise to the top of the list for a pharmaceutical company. So let's talk about addressing neglected diseases in the emerging and neglected diseases sense. So the Small Molecule Discovery Center is a partner, and we do a lot of work to support the Sandler Center for Drug Discovery at UCSF. And you already saw it, but this is their beautiful new logo. Advancing treatments for neglected tropical diseases. And there are two approaches that we've been taking with them to do this early stage drug discovery. One is a targeted approach, where we would have a biochemical target of interest, like CYP51, and do high throughput screening in medicinal chemistry, so that traditional mechanistic way of doing drug discovery. And then also phenotypic screening for whole organisms, taking advantage of our ability to do high throughput production of these organisms and high throughput analysis of the images that come out of these screens without having to understand quite so much about the biology and what makes these organisms deadly. So here are just two quick examples. This is Giardia, the little ghosties floating around in your GI tract, screaming boo. And this is Chagas disease. These are T. cruzi, this stain for kinetoplastids that live inside a mammalian host cell. So what I'm going to just have a few more slides on is doing phenotypic screening for schistosomiasis. So schistosomiasis is a very prevalent disease. 200 million people are currently infected across the world. The socioeconomic impact is second only to malaria among parasitic diseases. And there's only one treatment. I think one reason it's a particularly neglected disease is that praziquantel is quite a good drug. And so far, we don't have any resistance emerging. But when we do have resistance, we don't want to be caught sitting in the audience, right? I mean, because as soon as you get resistance, then it's going to be a big problem if you don't have a new drug waiting in the wings. There's several challenges to trying to treat schistosomiasis. One, you see that there are two stages of the life cycle. They have two different hosts, a mammalian host and a snail host. The adults are 1 centimeter long. They're sexually dimorphic, very complicated genetically. The babies, the somules, are still 200 microns, so they're also quite large. See a picture of them here. They don't propagate in culture. You have to have both of these animal hosts to grow the life cycle of the parasite. They're genetically heterogeneous, quite complex. And from a screening perspective, we're not just interested in them dying, but how they die, how long they die, what the mechanism of worm death is. Because one of the challenges is that some of the drugs and drug leads out there target one life cycle and not one part of the life cycle and not another part of the life cycle. And so we want to find drugs that target all the stages of the life cycle. And the state of the art for looking at these multiple phenotypes is visual inspection. So Connor Caffrey's lab in the Sandler Center have gotten very good at automating their visualization of these worms and scoring them for one of 6 phenotypes. But we wondered if we couldn't add more technology and computational power to this visual scoring so that we can do higher throughput analysis. So here's our team. Brian is in Connor's lab, Steve's in my lab, and Rahul Singh is a computational chemist, and he works in bioimaging, very complex image analysis at SF State. So we dealt with several of the critical bottlenecks to doing high-content imaging for schistosomials. First, Connor's lab figured out how to scale up the growth of worms. The baby worms are shed from snails, which you do by opening the curtain, and then the snails see the light, and then they poop these worms into the media, and then it's very quite exciting. These are the worms. And one of the biggest challenges is automating the dispensing of the worms, so we've solved that problem. Developing data collection protocols where we can monitor these worms by high content analysis, so automated microscopy in real time in the second time scale and over several days. Analyzing the time-dependent data, I'll just show you one teaser slide on that. That's the major area of focus now, and extracting quantitative parameters from this image stack. So here's an example of what those data look like. So I hope you can see these movies. Here we have a change in length over time. And so we're just measuring the delta length across the time scale of these movies as you add praziquantel. And this is what, I don't know if Connor named this, but I gave him a little R for the schistodisco. So as you see at high concentrations of praziquantel, you get hyperactive worms. But interestingly, at an intermediate concentration, you get these rounded up worms that move a lot less than the negative control. And you can monitor that across concentration and also across time. And you see this sinusoidal dependence. You go from normal amount of mobility to restricted mobility to hypermobility. Praziquantel is a neurotoxin. So this gives us one way of phenotypically characterizing these living worms before they die. Okay, so beyond chemistry, beyond screening, after we do screening, we need to do medicinal chemistry and chemical biology. Hi. Yeah, go ahead, take a seat. Okay, our judges are back in, so I'll wrap up quickly. So the medicinal chemistry group is very active in doing chemical biology and medicinal chemistry on materials that came out of the Sandler Center. For example, K77 is a compound that's moving into IND-enabling studies for Chagas disease, which is quite exciting. And we're working on identifying potential backups for this compound. Also, more chemical biology projects in targeted prodrugs for malaria. This is the active moiety, the trioxylane ring of artemisinin. And so this is a prodrug strategy for releasing artemisinin and another drug specifically into malaria and into other compartments that are rich in ferrous ion. And then another project in kinase target discovery and validation for T. brucei using a compound called hypothamycin, which covalently modifies only very particular cysteine residues in kinases. And so they've used a propargyl version, so a taggable version of hypothamycin to identify the target both by labeling this propargyl group with fluorescein and also by labeling it with biotin, doing a pull down and running an MSMS experiment to identify what we believe are 2 kinase targets in T. brucei of hypothamycin. All right, that didn't work. So something that's really important in academic drug discovery, do you see it's a major project. Any drug discovery project is really a major project and very difficult for any one group to do by itself. So we're very interested in building connections throughout UC and throughout QB3. And so just to give you an example, in case you didn't know, there are screening centers at all of the 3 QB3 universities. And we are differentiated in our services, and where possible, we partner and synergize. For example, around informatics, we have a common informatics system, and QB3 sponsors a computational chemist to help take that core informatics system and modulate it to be adequate for what the specific needs are of each of the 3 centers. So at Berkeley, you have a very nice screening center that focuses on siRNA and high-content imaging. That center works mostly with model organism genomes, so mouse genome and Drosophila. We have a human genome, and so we trade libraries. So you can do any genome at either site. And the Santa Cruz Chemical Screening Center focuses on small molecule high-throughput screening, and they have a particular expertise in marine natural products and in profiling infectious diseases. They run all of these natural products in their high-throughput screening library against a panel of, I think, 12 different infectious organisms. Okay, so we're now looking to expand throughout the UC system and talking to the far-flung universities about whether it makes sense to build pilot screening facilities that can then synergize with the larger, more established facilities that we already have in the Bay Area. Okay, so final slide. Why do we do lead discovery in academia? One, we can drive biological innovation in medicine by selecting targets and figuring out how to select patients. Small molecules teach us important things about biology. Chemical biology has been an invaluable tool in modern cell biology. And finally, and really importantly for this audience, our patience and our lack of a profit motive allow us and actually tell us that we should be working on neglected diseases and difficult problems. Okay, and this is our team. I hope you saw that. Okay, there we go. This is my boss, Jim Wells. Adam Rensler runs the chemistry group. Connor Caffrey and Rahul Singh, people whose work I talked about today. And thank you very much. You want me to give this to Kevin? Okay. I think this is like the Oscars, where you've got I guess. I don't know. Okay.
Just announce, oh, okay.
Okay, well, are there any, before he gets to the main event, are there any questions for me? Okay, I'll be around this after the presentations. Okay, thank you. So now our judges are ready to go.
Okay, so we're ready to go. So the audience vote came down in favor of the second proposal for male. And the judges' vote came out the other way for the first, which I think actually speaks to, I guess I'll just comment and I'll let the other judges comment, but I think speaks to the strength of both proposals. They were actually both very, very strong proposals. No obvious holes in either proposal. And in the end, I think we came down in favor of the first because it looked like it was a project that could make more progress quickly in the year that would be the prize for doing this work. Julia, do you want to add to that?
I agree with that. Both projects had a lot of experiments done already. I think the first project had really amazing follow-up. Having a compound already just enabled to show that the follow-up is feasible. I personally particularly like the NMR experiment. It was really nice to look at the mechanism. So there seems to be a lot of detailed information that can already be gained with experiments that are up and running. So no more development time is necessary.
Yeah, and I'll just, a few comments. The second proposal, again, it was a very strong proposal. One of the, perhaps challenges for that proposal will be it is a cell-based screen and probably more likely to produce false positives. But clearly, this had been thought through, and there were counter screens that would be in place to find the cream from the rest. Again, we put a lot of value in the fact that what in 1 year can you accomplish with these projects, and it just seemed that the first project a lot more in place. We would have been, I think, a little bit skeptical of the feasibility just because of the difficulty that the whole industry has had traditionally in blocking protein-protein interactions, and so the fact that a compound already exists and is structurally characterized was really key, I think, for us to pick that project.
Are there any questions for the judges? Thank you,
So congratulations to Jonathan and Andrew, and thanks both for participating. We're going to have a coffee break about 20 minutes, and we'll start again at 3:05. Thank you.